In this activity, we're going to look at the internal structure of a woody root and compare it to what you just studied in woody stems. You'll be working with a prepared cross-section of a Tilia or basswood root and identifying the major tissues that make up a mature woody root. Woody roots and woody stems share many structural features because both undergo secondary growth. That means both develop a periderm, secondary phloem, a vascular cambium, and secondary xylem. Even though roots and stems have different jobs, they're built using many of the same tissues. The outer protective layer of a woody root is the periderm, just like in woody stems. The periderm is made of cork cambium, and phloem. One important difference is that in roots, the cork cambium originates from the pericycle, not from the outer stem tissues, like it does in stems. As the cork cambium in the root continues to grow, it pushes outward and causes the cortex and epidermis to be shed. Over time, the periderm becomes the main protective covering of the woody root, replacing the original outer tissues. Continuing inside from the periderm, the main tissues are the secondary phloem, the vascular cambium, and the secondary xylem. Just like in woody stems, woody roots also contain xylem rays and phloem rays. These rays run radially from the center outward and allow materials to move side to side across the root instead of only up and down. In the very center of the root, you'll also see remnants of the primary xylem, which formed earlier when the root was still young and herbaceous. Before we get started with the activity, answer this question. Why do woody roots and stems share many structural features, and how might these features differ based on their location and function in the plant? As you think about this, consider what roots and stems both need to do in terms of support and transport. Then think about how their different jobs, roots in the soil and stems above ground, might cause some differences in how those shared tissues are arranged or emphasized. Before you start drawing, take a moment to visually walk through the root from the outside inward. The periderm is the outer protective layer. Just inside that is the secondary phloem, followed by the thin ring of vascular cambium. Inside the cambium is the large region of secondary xylem, and in the very center you should see primary xylem. Look for xylem and phloem rays running radially across these tissues. As you look at the image, you'll make a sketch in your lab notebook and label these structures. Use their relative positions, what's outermost, what forms a ring, and what's in the center to help you place and label everything correctly. Answer the following compare and contrast questions. First, how does the arrangement of tissues in a woody root compare to that in a woody stem? Think about what tissues are present in both and whether their order and proportions look similar or different compared to the stem you studied earlier. Second, are growth rings visible in woody roots like they are in woody stems? Why or why not? Think about what causes growth rings to form in stems and whether roots experience the same seasonal growth patterns or environmental conditions that produce those visible rings. Since it looks like the woody root we looked at was younger, I found a slide of an older basswood root. This activity helps you see that woody roots and stems are built using a similar developmental plan, even though they serve different roles in the plant. It reinforces how secondary growth works and how plant structure reflects function, while also strengthening your ability to interpret real anatomical cross-sections.